recorded videos, assuming there are no issues. Okay, human blood. Blood is our transport system in our bodies. We use blood to carry nutrients, to carry oxygen, to carry white cells to fight infection, and we use it then to remove carbon dioxide from our cells, and we use it to get rid of waste products that cells form. And this shows you red blood cells moving through our capillaries. We see that there are millions and millions of red cells present here, and we see that they really fill up the space. So what is it about blood? Well, blood is red. It's red because of the presence of the hemoglobin. It is sticky. It is opaque, meaning you can't see through it. If you've ever tasted your own blood, hopefully only your own blood, you'll notice that it has a coppery taste, a metallic taste to it. The pH of the blood is right around 7.4, somewhere between 7.35 and 7.45. It is slightly alkaline. Its temperature is above our normal temperature. It's at 38 degrees instead of 37 degrees. The amount of oxygen present in the blood determines how red it is. The higher the oxygen concentration, the brighter red it gets. So when we're bleeding from a cut or a scratch, if it's dark red, it's usually venous blood. Hopefully it's venous blood. And if we're bleeding from an arterial injury, it's going to be bright red. And how much blood do we have? Well, we don't have enough to fill up every vessel in our bodies. The average volume in males is somewhere between 5 and 6, and with females, it's 4 to 5 liters. This is all based on the differences in average body stature. So what does blood do for us? Well, it carries oxygen. It carries nutrients. It picks up wastes. It transports hormones around from the gland that secretes them to their target cells. It regulates our temperature. It regulates our pH. It regulates our fluid volume. How much water do we have in our plasma? Is our plasma diluted because there's more water, or is it more concentrated because there's less water? And it protects us. It helps us fight infections because we have the white blood cells that are the infection fighters. We also have fibers in solution that will plug a hole, as well as the platelets. The platelets will also plug a hole if we're losing blood. So, transport functions: deliver oxygen and nutrients to the blood cells, take the metabolic waste from the lungs and from the cells themselves to the kidneys for removal, and metabolic waste too, like carbon dioxide. That's a metabolic waste that we get rid of in the lungs. We breathe that out. The nitrogenous ammonia-based waste that we generate in the breakdown of proteins, we release those through our kidneys as urine. And of course, we transport hormones from the endocrine glands to their target cells. We regulate our body temperature. We absorb heat. We distribute heat. On a cold day, we would restrict the flow of blood to our skin so that we don't lose heat. On a hot day, we would increase blood flow to the skin so that we can radiate heat. We maintain the pH of our blood through the use of a mechanism known as buffering. We use sodium bicarbonate to neutralize acids so that we don't become too acidic or too alkaline. And we always keep enough fluid in our circulatory system to allow us to move our blood cells, because blood cells have to have some sort of medium to float in, and that's what the plasma does. And I mentioned this already. How do we protect? Well, we have platelets in our blood that will plug a hole. It's called a platelet plug. Platelets are cell fragments, and they will come together to form a plug to fill an injury, damage to the body. We have plasma proteins, such as fibrin, which is in solution, which will solidify when activated to form a large blood clot. And of course, we carry antibodies to glom onto foreign cells with their antigens. We produce complement proteins, which will destroy cells or inactivate pathogenic cells. And we have the white blood cells, which are the true infection fighters. Those are the only blood cells that actually kill other cells. We have white blood cells, which are called natural killer cells. And that's what they do. They hunt down pathogenic bacteria. They hunt down infected cells. They hunt down cells that contain that have, that are infected by a virus and destroy them. Anything, a cell that's worn out, the white blood cells will destroy those. This shows you white blood cells going after a bacteria. You can see the bacteria is a tiny little, diplococcus there, two bacterial cells together, and the white blood cell will go after that, engulf it, and destroy it. And there it goes. Right there. And once it's inside the cell, the lysosomes will break it apart. Blood is the only fluid tissue in our bodies. It is connective tissue because it has living cells in a matrix with fibers. The matrix is the plasma. The living cells are the red cells and the white cells, and the cell fragments, the platelets. And the fibers are the fibrin proteins that are in solution that, when activated, will form solid fibers. So it meets the definition of connective tissue. The cells in our blood are called the formed elements, and they are suspended in the plasma. The formed elements are the erythrocytes, or the red blood cells, the leukocytes, the white blood cells, and the platelets, which are just cell fragments, but they have a very important role to play. The only cells that are complete, however, are the white cells. The white cells are the only ones that have a nucleus. Red cells kick their nucleus out during their development. Platelets are nothing but cell fragments. The white blood cells have a true nucleus. Now, how long do these cells last? Well, most only survive for a few days. Some last for up to 4 months. Red blood cells will go for 4 months. The platelets last for a few days. Some white cells can last for years. Most only last for a few days, but some white cells will last for years. The blood cells all come out of, or rather most of them come out of the bone marrow, in what we call the red bone marrow, and they generally don't undergo mitosis on their own and cell division to reproduce. They're continually being made in the bone marrow. Now, if you take a tube of whole blood, since it is a suspension, you can get it to separate. Remember what a suspension is? It's an opaque fluid with substances held in solution in the fluid, held in place, and you can't see through it. That's a suspension. If you were to take a tube of whole blood and place it in a centrifuge and spin it down, you would move all the red cells and the white cells and the platelets to the bottom of the tube, and you would have the plasma on the top. So about 55% of the of whole blood is made up of the plasma. You would have a very, very thin layer of white on top of the red cells. That white layer is called the buffy coat, and it's made up of white cells and platelets. And then the rest of it is made up of red cells, the erythrocytes. The bottom 45% of the tube would be red cells, and it would look something like this. You take a sample of blood, place it in a tube, place it in a centrifuge. That's what this thing is right here. Spin it down. The heavy substances, heavy particles, move to the bottom of the tube, and the light stay at the top. That means the plasma stays on top. And so it separates out with plasma on top of the cells. 55% of it's plasma, 45% of it is red cells. There's a very thin layer called the buffy coat, which are the white cells and the platelets. And it looks something like this. You see the whole blood on the left, the yellow straw-colored plasma on the right side with the very thin buffy coat layer and the red cells underneath that. So plasma, what is it made of? It's about 90% water. 90% water, and the rest of it are proteins and salts and nutrients and wastes and buffers. The proteins include the albumins, same protein that you encounter in egg white. The globulins are part of our infection fighting. The globulins actually make up the are like our antibodies. Fibrinogen, which is the inactive version of fibrin, which is our clotting protein. Prothrombin, which activates fibrinogen, and then the enzymes and the hormones. And of course, then you've got the sodium and the chlorine, and the potassium and magnesium and calcium, and you have glucose, and you have ammonia, and you have sodium bicarbonate, and you have carbonic acid. One of the terms we use to describe a measurable concentration in our blood is called the hematocrit. The hematocrit is a measure. If you take a sample of whole blood and spin it down, hematocrit is a measure of the number of red blood cells, or the percent of red blood cells in whole blood. And so when we spin that down and get that sample, and we see that the top 55% is plasma and the bottom percentage is red cells, we generally look for something in the value of about 40 to 55%. 45% is a good number. 46% is the average. We simply it's a very simple measure. What percent of the test tube is filled with red cells? And we're looking somewhere around 45% in males, 42% in females. The difference owes to the presence of estrogen in the female. Estrogens don't stimulate red cell production like testosterone does. And so we have a slightly lower red cell count in your female patient. Hematocrit values can tell you a lot of things. If the hematocrit is low, it may be suggestive of anemia. Now, anemia is not a disease unto itself. Anemia is a symptom of a condition. But if a patient has a low hematocrit, we would say that they are anemic. They don't have enough red cells. Now, why is that of a concern? Well, the red cells carry oxygen. And if the red cells are carrying the oxygen, if we don't have enough red cells, then we don't have enough oxygen being carried around to our bodies. And so we're tired. We don't have energy. We don't think well. Our cells are not getting the oxygen they need to release ATP. If you remember cellular respiration and AP1, what is the role of oxygen? Oxygen grabs the electrons coming off the electron transport chain. Without oxygen, those electrons don't come off the electron transport chain. The chain backs up and we don't make any more ATP, which is what we use for energy. So low oxygen means that we are not getting enough energy. And if we don't have enough red cells, then we're not going to be carrying enough oxygen. So if our hematocrit is low, which is the hematocrit is the measure of, is the percentage of red cells in our whole blood. If that's low, it might be indicate anemia. If it's high, if it's elevated, it can indicate a condition known as polycythemia, where we have too many red cells. And this is one of those conditions where if some is good, more is not better. Because if we have too many red cells, then we are crowding out our white cells. Just because we have more red cells and theoretically could carry more oxygen, that's not what happens. We have so many extra red cells that we start blocking the vessels. We start generating clots and we start blocking blood flow. So it is, while it sounds like if some is good, more is better, again, this is one of those cases where if some is good, more is not good. So this is one of the ways we can conduct a hematocrit. This is what's known as a microcentrifuge. And you take a very small sample of whole blood in a thin, what we call a capillary tube, and we place that in the centrifuge and spin it down. And then we can measure the percentage of blood fairly accurately in here to give us the hematocrit. Because the hematocrit is a very useful diagnostic, parameter, if you will, result. If we know the hematocrit, we can say, well, it's elevated, it's low, it's normal. We can say, are we anemic? Are we, what would say a hypercoagulable state, meaning we have too many red cells and we're going to start forming blood clots, or are we fine? So a little what we call a capillary tube is filled with blood, and it's put in the centrifuge and spun down. And we have, just like I showed you in the bigger test tube, we have the plasma with the buffy coat layer and the red cells. And then we can measure the percentage of red cells in that tube. We have what's known as a microhematocrit reader. We don't have to get a big test tube to do this. It's a very simple device. You place the tube in this reader, and you simply look at the layer, where you look at the layer that's the top of the red cells. Excuse me. And you read the number off. In this case, this lines up to a hematocrit of somewhere about 80%, which is definitely of concern. But suppose this line, when you lined it up, was at 30%. That would also be of concern. We want to see our numbers right here in the middle, about 45%. And I know technology has replaced a lot of this. Technology can take the tube and evaluate it very quickly. And there is a certain level of subjectivity in using these mechanical devices, because everybody sees things a little bit different. And you're depending on precision, because precision is important. But even these devices, these older, much older hematocrit readers, simply involve placing the hematocrit tube in this bracket here, and then raising this lever until you're lined up with the top of the red cells, and you get your number in here. And it still works. It's not probably as precise as technology, but it still works. Okay? Because your level of concern, if you're 46% or 45%, that's not a very significant difference. If you're 30% as opposed to 45%, that's significant. And this older technology will still detect that for you. New technology gives you a much more precise answer. Okay, this is another type of test. When we're evaluating blood, we need to look at things like the hematocrit. We need to look at things like the amount of hemoglobin that we're carrying in the blood. We need to look at the number of red cells and white cells that are present in the blood. This test here is to determine what we call sedimentation rate. Hematocrit tells us what percent are red cells, and what percent's plasma. Sedimentation rate is an indicator of some sort of infection or inflammation going on in the bodies. We call it the erythrocyte for red cell sedimentation rate, or ESR. We abbreviate it as the SED rate. It's a measure of how fast red cells sink to the bottom of a specific tube in an hour. It allows us to see if some sort of inflammatory response, a response to infection, is going on. It doesn't tell us where the infection is. It doesn't tell us what kind of infection it is. It doesn't even tell us where it is, but it does tell us that something's going on. So when you're looking at evaluating your patient, you actually are looking at the big picture and trying to figure out what's going on with them. You always want to start your diagnosis in a very broad approach. You look at things in a very broad manner, and then you start narrowing it down. Just because they have a pain in their lower right side, for example, doesn't mean that they have appendicitis. It can mean a whole lot of things. And sometimes if they have a pain in their upper right side, doesn't always mean it's their gallbladder. There's all sorts of things that can cause that. So we know that we can't assume. So we have to have a very broad approach. Sedimentation rate will tell you at least, is there inflammation? Is there an infection going on somewhere? So what we do is we take blood and we place it in a tube known as a Westergren tube, and then we measure how rapidly it falls. If there is inflammation going on, what will happen is the blood cells will start to stick to each other. The fibrinogen, the plasma protein in here, the plasma protein starts to get activated during an inflammatory response. Now, an inflammatory response in our bodies is both a good thing and a bad thing. Localized inflammatory response, we have those all the time. If you have a cut or a scratch on your skin, the area around the cut or the scratch is red. It's red as a result of the inflammatory response. When there is this inflammation, a localized inflammation, it means that you've increased the red cell, you've increased the blood flow to a region, more red cells are going in to bring in more oxygen, more glucose is coming in to feed cells, and more white cells are coming in to kill bacteria and take out dead cells. It's all part of the healing process. But if you have a massive inflammatory response going on, it usually indicates there's a major infection going on in your body somewhere. So when we have an inflammatory response, our red cells tend to stick together a little bit. And when they stick together in a very specific pattern. They call it the rouleau formation. They look like a stack of Pringles, if you will. And so these cells, when there is an inflammatory response going on, our red cells are going to stick together, clump together, and they're going to settle out in this tube faster. These are the rouleau formations. They stack together, they look like coins. You stack up some coins and you knock them over. Or if you open up a can of Pringles and you pour them out all at once and they all come out, stacked, sort of like they do in the picture here. This is what our red cells do, because if there is a major inflammatory response, the fibrinogen, the plasma protein that controls our clotting, is going to start to become activated, and you're going to start seeing these red cells stick together, and they look like this. These are rouleau formations. So if we do a sedimentation rate test on a patient and the sedimentation rate is rapid, the red cells sink to the bottom very quickly, that indicates that something's going on. May not know what's going on just yet, but now you've got a clue to point you in hopefully the right direction. And you can even see this under the microscope. I've got some slides later on that I'll show you just how rapidly or how significant this rouleau formation looks like under the microscope. I mean, this is nice. This is but this is a stylized drawing here. The reality is, it is pretty significant too. So, okay, another thing we would look at here. Well, yeah, this isn't bad. Something like this. Let's take a look now. Let's go on. So we've seen sedimentation rate. We've seen hematocrit. Now another measure is hemoglobin. Hemoglobin is the protein, the quaternary protein. If you remember back to AP1, hemoglobin is the quaternary protein that carries oxygen for us. It's what makes our blood red. Hemoglobin in the presence of oxygen turns red. And so hemoglobin carries oxygen to our cells, where it diffuses out across the cell membrane and supplies oxygen to the cells, and carbon dioxide diffuses from the cells into the hemoglobin, and we carry it back to the lungs. And usually when we have a complete blood count, what we call a CBC, you go to your doctor's office and they send you to the lab because we want to have a complete blood workup on you and see what your hemoglobin level is. And so we detect from the blood sample, we get the results, and says your hemoglobin is X percent or X grams or whatever, and that gives us an indicator of do we have enough hemoglobin or not enough hemoglobin. Okay. Hemoglobin, it's one of those grams per deciliter, like or milligrams per deciliter, like we measure blood sugar. So how many, what's a normal level of hemoglobin? Well, in males, it's based on average body stature. It's somewhere between 14 to 18 grams per deciliter. 14 to 18 is a good range. In females, it is 12 to 16. So there's a lot of overlap there too. As males age, the hemoglobin levels tend to drop off a little bit. In average, they somewhere between 12 to 14, 12 to 15, from 14 to 18 to 12 to 15. And females also see a decline in their average value for hemoglobin, 11.7 to 13.8, but somewhere, 15 is a good number. 15 grams per deciliter is a good number. 15, 14, somewhere in there. These are good values. You get below 13, you have some concerns. You get below, if you're, so you start paying attention to those values. Now, there's a couple different ways to measure hemoglobin. The best way, of course, is, analytically, the blood goes off to the lab evaluates it analytically, and you get a value. There are other approaches that are a little more subjective. This is what's known as the Tallquist scale. This was used for years. We only point this out to you now to show you what was used to be done. Here you would take a sample of blood and place it on a piece of what we call blotting paper. It has a high fiber concentration. It means it holds the blood in place better. Now, if you look on the right-hand side of the picture, you see a series of red bars, different shades of red, from light orange all the way to scarlet. And those little circles there are actually holes in the paper. And what you would do is you would put a drop of blood on this blotting paper and slide it underneath the page till it lines up with the hole in the page, and you can match up the concentration and say, well, this is the percentage of hemoglobin from this blood sample. Now, a lot of things wrong with that. First of all, it depends on the subjective opinion of the evaluator. Second of all, the color of the blood on the blotting paper changes as it dries. And so you're sliding that paper along up and down underneath that page with the holes in it, trying to line it up, and the color's changing over, if you don't do it right away, your color is going to change as the blood dries in the paper. It works for what we would say as a quick and dirty test. It would give you something, a subjective value, because then again, everybody would look at this differently. Everybody sees these values differently. It's like, if you take it and place it underneath the second one down, somebody may read it, oh, that's too dark or too light. So it is very, very subjective now. And the scale here is pretty broad. This next, the one at the top gives you a hemoglobin reading of 15, and the one below it gives you a reading of 14. And that may be a significant value. So we don't do this anymore. It's not objective. It's not very analytical. So, but it's, I can't imagine anybody doing it, but it still works, just not as precise. So, okay, now let's move on. Okay, so now we know that we can measure hemoglobin, we can measure sedimentation rate, we can measure hematocrit with our blood samples. Something else we absolutely need to know are the blood types. Human blood has a wide variety of blood types. There are 4 big ones, the 4 big categories, but there are about 90 subcategories altogether. And our blood type is genetically determined. It's genetically determined from we inherited it from our parents, and so that makes that, and that becomes very significant, particularly if we're talking about Rh factors, which make our blood positive or negative. Now, the big 4 belong to what we call the ABO group, where we're either A, B, AB, or O. And then we consider, are we Rh positive or Rh negative? Most people are Rh positive. But it's one of those, yes, it's a genetic factor. Does it affect us? Only when we get transfusions, whether we're Rh positive or negative. What forms us? What makes us Rh positive or negative? What makes us AB, or O are the presence of antigens on the surface of our red cells. Now, those antigens are the markers that tell us what the cells are. They're also the markers that tell our immune system that we are us. These are the glycocalyx structures that we talked about in AP1 in the cell chapter. These are the markers. They are they're a carbohydrate and a protein marker that is unique to that cell, telling us it is type A or type B. It's also unique to us, so that our immune system recognizes it. These glycocalyx structures are the antigens that identify us. Now, in the ABO system, there are only 2 antigens, A and B. If you have type A blood, you have the A antigen on the surface of your red cells. If you are type B blood, you have the B antigens. Now, if you are type A blood, your enemy is type B. You see, when you're type A blood, you have the A antigen, but you also have antibodies. Antibodies are proteins that our blood makes. These antibodies will go out and look for foreign antigens, so they can destroy them. These are the antibodies. You know, antibodies are a good thing when we're exposed to a foreign pathogen like a bacteria or a virus. The bacteria or the virus has their own antigens on the surface of in the case of the virus, it's on a protein coat. In the case of bacteria, it's on the cell wall of the bacteria. It identifies the bacteria as bacteria. Well, our bodies can determine that bacteria is dangerous or foreign because it's a foreign antigen. The glycocalyx structure sticks up and says, we say, that's foreign. And then we have antibodies which will go attack it and destroy it. So if you are type A blood, you don't want to have B blood. You don't want to be exposed to B blood at all. So if you're type A blood, how would you know that B blood was introduced to you? If someone forced you to have a transfusion, or if you got a transfusion, how would you know that you were given the wrong blood? Well, because the foreign blood, if you're type A, you have blood with A antigens on its surface. You would also carry what are known as antibodies, not for your own blood, but for foreign blood, different blood, foreign meaning different. So if you're A blood and you have A antigens on your red blood cells, you're going to have B antibodies. B antibodies will attack B blood. B antibodies will attack B antigens. Red blood cells, type B red cells, are going to have B antigens. Just like you have A antigens on your red cells because you have A blood. You have A antigens, but you're going to carry B antibodies. That's what this first line says here. If you're group A, you have A antigens and you have B antibodies. You're not going to attack yourself, but you're going to attack any other blood that comes in that has those antigens, those antibodies there. I'm sorry, those antigens. So you'll have A antigens and you'll have antibodies which will attack B antigens. If you're type B, you've got B antigens. If you were type B and someone gave you a bag of A blood, then you have A antibodies. You would see the A blood as foreign because the A blood has A antigens. It's like the A blood has little flags on its surface saying, hey, we're the A antigens, we're foreign. And you'd have your A antibodies to attack it. I know it gets confusing, but if you're A blood, you've got A antigens. If you're B blood, you have B antigens. If you're A blood, you have B antibodies. Your antibodies are always the opposite of the antigen. You're not going to attack your own blood, but you're going to attack what we would consider foreign blood. Now, so group A, group B. If we were in class, this is the point where I'd say, how many know their blood type? And of those that know their blood type, how many are A, how many are B, A, B, or O. A is probably the most common, right behind or either slightly ahead of O, or A or O are the most common types. Anyway, the third category, group AB. AB has both A antigens on its red cells and B antigens. So if you're AB, you have both sets of antigens, but you have no antibodies for your blood, because if you did, you'd be attacking yourself. So AB has the antigens and no antibodies. Type AB blood is considered the universal recipient because with no antibodies, they can get blood from A, they can get blood from B. Not going to get a reaction. So they're not going to respond. They're not going to, someone who is AB can take anybody's blood. No antigens no antibodies to attack it. The fourth category, O, has no antigens on the surface of its cells. It does contain A and B antibodies, but it has no antigens. Now, what does that mean? It means if you are type O blood, you are the universal donor. Because your blood has no antigens on it, you can give to anybody. AB is the universal recipient. It has no antibodies, so it can take blood from anybody. Group O has no antigens. It can give to anybody. Someone who has O blood will not cause a reaction in anybody. So AB is the universal donor, O is the universal, I'm sorry, AB is the universal recipient, and O is the universal donor. If you have an antigen antibody reaction, we will end up with clumping. That's what agglutination means. If you have A blood, if you're normally A, type A, and you are given B blood, this is where it gets very serious. The foreign antigens on the B blood will attract all your antibodies, and they will glom on to those red cells, and they will start clumping together and start forming clots throughout the body. And the reaction is so severe that it can be fatal within hours. So we definitely want to avoid any kind of transfusion reaction. So we don't, if you're type A blood, we don't want to give you B blood. If you're type B blood and we gave you A, it would be the same result because the A blood has A antigens. You have A, if you're type B, you got A antibodies. You attack that blood, it clumps together, and you get all these clots forming throughout your circulatory system, and you could die very, very quickly. We go to great lengths to avoid problems with transfusions because if you've ever been in the hospital, you know that if you have an IV line going into you, that there's a whole series of numbers on the IV, and that matches up with any bag of blood or bag of medication that's hanging on the line. Those numbers have to match, and when they come in and hang that unit of whatever on your IV line, they're going to read off the numbers, and a lot of times they'll ask you to look at the number and pair them up or read it off and check it. They'll double and triple check to make sure there are no mistakes, because you don't want to give someone the wrong blood. Now, there is one advantage to this this whole mechanism, is that there is some, because of the universal donor, type O, we can give O blood to anybody without a reaction. A person can get O blood and will not have any reaction. Doesn't matter if they're type A or they're type B or they're type AB, you can give them O blood with no problems whatsoever. So generally, in an emergency room or an emergency department, you're going to keep lots of O blood around because you don't necessarily have time to do what they call the types of blood and cross match it to find a donor or find blood in the shelf that is available to use. So you use type O. Now, there is one other complication in here, and that is the RH factor. The RH factor is based on the presence or absence of another antigen. We have antigens that tell us type A and type B. Now we have an antigen called the D antigen. If you have this D antigen, then you are considered to be Rh positive. If you don't have the D antigen, you're Rh negative. But there are no, normally, if you're Rh positive, you have no antibodies. You have no antibodies for the Rh factor because you've got everybody, if you're Rh positive, you have the D antigen, you don't have antibodies. However, and this is the however, if you don't have the D antigen, in other words, if you are, you're Rh positive, you have the D antigen. More people are Rh positive than are Rh negative. People that are Rh negative, individuals that are Rh negative, don't have the D antigen. They also don't have antibodies. So it shouldn't be a problem. The only time that an individual would have antibodies is if they are Rh negative, meaning they don't have the D antigen. But they are exposed to Rh positive blood. So you have a patient that is brought into the emergency room and they're bleeding profusely and they are, A negative. And so you hang a bag of O blood on them because O is the universal donor and you don't have to worry about, matching it because O never causes a reaction. Except, what if that blood's O positive? If that blood is O positive, the presence of the D antigens will cause the formation of antibodies so your patient's given a bag of O blood. That's great. That's protocol. They're injured, they're bleeding out. You give them a bag of O blood, but a mistake was made. And instead of O negative blood, it was O positive blood. The O positive blood has the D antigen. Well, your patient's never been exposed to positive blood before, and they start forming antibodies. They start forming antibodies to that positive antigen. The antibodies build up and build up. By the time the antibodies build up to a level where they would do something and attack the O positive blood, the O positive blood has disintegrated. See, red blood cells that are stored in a blood bank will only last, if they're frozen and sold out, will only last for about a month. So that doesn't, even if you give a fresh transfusion from one patient to another, it's going to take quite a while for your patient to develop antibodies to those foreign antigens. So so what you do is the ideal approach is you give your patient blood that's not going to cause any kind of reaction. O blood is the universal donor. No antibodies to attack anything. No antigens to attack anything. You also give them, make sure it's O negative. So there are no antigens from A, there are no antigens from B, and there are no Rh antigens. So the ideal blood to give to an injured victim is O negative blood. But mistakes happen. You accidentally give them O positive blood because the blood bank in the hospital sent the wrong batch to the emergency room. Hey, it's all red. It's blood's red, so it ought to work. Well, so they sent down O positive. So you give your O positive blood to an O negative individual, or to an A negative individual, or to an AB negative individual, or a B negative individual. Okay, no problem. They've never been exposed to positive blood before. They build up these antibodies, but now they've been sensitized. Now, this is not a problem if they are never exposed to Rh positive blood again. But if they are exposed, if they accidentally get another transfusion of the wrong blood, then they would have a life-threatening agglutination clotting reaction that may kill them. There is another problem that can develop. Well, let me get back here before I go on to this. There is another problem that can develop, and that is if you have a pregnant female who is Rh negative and the father was Rh positive. Now, Rh positive, as far as a genetic trait goes, is a dominant trait. The absence of the D antigen, Rh negative, is a recessive trait. The dominant trait always wins. So if the father is Rh positive and the mother is Rh negative, the baby will always be Rh positive. If the mother is Rh positive and the father is Rh negative, the baby will still be Rh positive. It doesn't matter who has the dominant trait, doesn't matter who has the Rh positive trait, that, the Rh positive trait always wins. So, but if you have an Rh negative mother and an Rh positive father, the baby will be Rh positive. Now, what will happen is there is no mixing of blood across the placenta, no mixing whatsoever. So we're not sending the mother's blood, it's not going to the baby's blood. The baby's blood's not going to the mother's blood. However, at delivery, there will be the opportunity for those for the blood. Let me rephrase that. During delivery, there is the opportunity for the mother's blood to become sensitized to the Rh positive antigens. Nothing's going to happen because her exposure to any Rh positive antigens will not cause her any harm. However, she will also become sensitized to the Rh positive antigens, and that may not be a problem whatsoever, except if she gets pregnant again. Rh positive father and she's Rh negative. Now she has Rh positive antibodies. Those antibodies can cross the placenta and attack the developing baby in utero. So the approach is to use a drug called RhoGAM. RhoGAM destroys Rh positive antibodies. And it's a standard protocol. If the mother is Rh negative and she's pregnant, it doesn't matter if it's her first baby or her 10th baby, then the OBGYN will routinely prescribe RhoGAM to destroy any possible Rh positive antibodies. And so, if you're an Rh negative female, you can be pregnant as many times as you desire, and you shouldn't have a problem with this. Okay. The only way an Rh negative individual can develop antibodies is if they are exposed to the Rh positive antigen. So other than pregnancy, it's pretty difficult for an Rh negative individual to become sensitized. Let's go back here. Oops, what did I do? Didn't want to do that. Let's get. Okay. Now, the types of blood, the formed elements. How many do we have? This is in a cubic millimeter of blood. Erythrocytes, 4 to 6 million. 5 million is a nice, good round number to remember, about 5 million per cubic millimeter. The leukocytes, the white cells, between 4,000 and 11,000. 5,000 in a cubic millimeter is a good number. So 5 million red cells, 5,000 white cells. Now the order of the types of cells, we have the neutrophils, the most common white cell, the lymphocytes, the second most common white cell, the monocytes, the third most common, the eosinophil, the next to last, and the basophil, the last. And then we have the platelets somewhere around 500,000. So you can go 5 million, 5,000, 500,000. Now, how do you remember the order of these white cells? There's a little mnemonic, a little memory aid for there. We have neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And I'll show you that mnemonic here in a second. Here's what they all look like. There are the red cells right there. They don't have a nucleus. They stain, they show up on a blood smear as a pale red or pink cell. Lots of them in a typical blood smear. The platelets show up generally as little purple structures. They're nothing but cell fragments. The white blood cells show up with their nucleus, very visible, stained dark purple. The cytoplasm usually stains light purple. It has to do with the dye that's used when you do blood samples. So there's the neutrophil. The neutrophil has a 3-lobe nucleus. There's another neutrophil over there. You can't tell, but it also has 3 lobes. The lymphocytes, the second most common, have a very large nucleus that almost fills the entire cytoplasm. The third category are the monocytes. They have a nucleus that looks like a kidney bean. That's a monocyte right there. The eosinophils, the next to last common, has a multi-lobe nucleus, but it only has 2 lobes. Eosinophils tend to show up when we have a parasitic infection like worms. Like your patient has a tapeworm and their blood work, you don't know, they're losing weight, they're tired, what's going on. You do their blood work up and discover there's a whole boatload of eosinophils when there shouldn't be, because they show up in parasitic infections, they show up in allergic reactions. They have a 2-lobe nucleus. And then the last one, the basophils, they look like blackberries. They don't have a very distinct nucleus, and they have lots of, the way the stain takes in here, little round lumps, it looks like a big blackberry. So erythrocytes, 5 million erythrocytes, 5 million red cells in a cubic millimeter of blood. If you look at a blood smear under the microscope, you would generally see a whole bunch of red cells and 1 white cell in your field of view, and that would be normal. These are all red cells up here. You can see platelets. Remember, you have about 500,000 platelets in a cubic millimeter versus 5,000 red cells. So you'd see one, maybe 2, maybe 3 white cells, but that's all. And you'd see lots of red cells. This is what an erythrocyte is. It is what we call a biconcave disc. It has a dimple on both sides of it. It's a flattened disc in the middle that increases the surface area for picking up and releasing oxygen. And it is, these things will last for about 4 months in our blood, and then they go to the spleen to be broken down to recycle the iron and then break the hemoglobin down. These are the platelets. Platelets are cell fragments. These little purple dots are the platelets. About 500,000 platelets in a cubic millimeter. We have, their role is to form a plug when there's a penetrating injury into the blood vessel to stop bleeding. Now, the white cells. This is the mnemonic. Never let monkeys eat bananas. N for neutrophil, L for lymphocyte, M for monocyte, E for eosinophil, and B for basophil. This will always tell you the order of the 5 white cells. So there's 5 different types of white cells. Neutrophils are the most common, basophils are the least common. Neutrophils, N, lymphocyte, L, monocyte, M, eosinophil, E, and basophil for B. Never let monkeys eat bananas. I have no idea what we've got against monkeys, but the mnemonic works. Okay, neutrophils. They generally have 3 lobes in their nucleus. They show up right away during an infection or an injury. You'll have lots of neutrophils showing up to start the process of killing bacteria and start the healing, the healing and recovery. So just see, you can see multi-lobed, multi-lobed, multi-lobed. Again, more neutrophils in here. Now there's something else going on here. These are the rouleau formations. See how the red cells are stacked on top of each other up here at the top and on the sides? You see that they're clustered. They're not supposed to look like that. A red cell is supposed to look like this, just scattered randomly. They're not supposed to be in contact with each other. Let's go back a little further, or look like this. These are these gray things, which are really red cells. They're not stacked on top. But what we're seeing here is that we have the stacked structures. We also have 4 white cells present, 4 neutrophils present. In a slide, in a field of view that shows you this many red cells, you should see 1, no more than 2 white blood cells. And here we see 4. That the fact that we see 4 white cells and we see rouleau formation in the red cells tells you that there's something going on from this slide. There's rouleau formation. It means the sedimentation rate is high, and we see 4 white cells. There's probably an infection going on where this blood sample came from. Lymphocytes. Lymphocytes are the white cells. Second most common type of white cell. They have a very large nucleus. These are the infection fighters. They're either going to be T cells or B cells. T cells mature in the thymus, B cells mature in the bone marrow. The T cells are the natural killer cells. These are the cells which destroy other cells. They kill infected cells. See, and the nucleus is so large, it almost fills the cytoplasm. That's how you can tell. Look for an edge of cytoplasm visible around the outside, but it's a very large nucleus. Monocytes. Monocytes have a kidney bean shape or a horseshoe shape of their nucleus. They're the largest white cell. They become the macrophages that will destroy bacteria and viruses, and they'll show up at an infection to kill infected cells and remove damaged cells. They will immediately move into the tissues from the site of an injury to go after the bacteria that might be present or to help repair the damage. Very distinctive shaped nucleus. A kidney bean, a horseshoe. That's how you perceive it. Okay. This is a monocyte. There's a very distinctive kidney bean shape. Third most common type of white blood cell. Very hard to spot because you're going to see nothing but neutrophils and lymphocytes. Now, this is the eosinophil. This is your number 4 type of white cell. The nucleus is got 2 lobes. It's easy to confuse this with the neutrophil because the neutrophil has 3 lobes. Eosinophils show up during allergic reactions and parasitic infections. So If you see a lot of eosinophils when you do a blood smear, that tells you something's going on, because you don't normally see a lot of eosinophils. So again, there's one, 2 lobes, very distinctive. There's your eosinophil right there. And your last category is the basophil. The basophil has a completely filled cytoplasm with its nucleus. The nucleus looks like lots of little purple dots. It looks very much like a blackberry here. Now, here is one type of pathological condition. This is a type of leukemia. Now, a couple things you see here. First of all, the red cells have formed such very dramatic rouleau formations, to the point that the red cells are clumped together, almost useless. The second thing is that you see many, many, many white blood cells. Leukemia is a malignancy. It's a cancer of the blood caused by overproduction of red cells. I'm sorry, overproduction of white cells. The white cells can't function because they don't mature. The red cells can't function because their inflammatory response is to cause them to stick together. So here, if you look, you see all these different white cells in various stages of rapid growth. Like this is a 3-way cytokinesis here. Here is what might be a monocyte and another monocyte. But here we see, there we go. Up here, there's very rapid cell division going on right here. You can see they're in the last stages of telophase and cytokinesis. You have lots and lots of imperfectly formed white cells that aren't going to do anything. This is what we have acute leukemia. Acute leukemia is more dangerous than chronic leukemia. Acute leukemia has sudden onset and grows rapidly, and it has a higher mortality rate than chronic leukemia. Both of them, can be treated very successfully. The cure rate for leukemias are well into the 90% now, but acute leukemia tends to be a little more dangerous than the chronic leukemia. Both of them involve overproduction of white blood of white cells, but see, they're not differentiated into the various types. They all look indistinct. Can't really tell what they are here, and so they don't fight infections. Your patient with leukemia is going to be prone to infections from every bacteria that comes along, and they're going to be tired. They're going to be extremely sluggish because their red cells can't carry enough oxygen to keep the other cells happy. Red cells are all clumped together in these giant rouleau formations. Atypical leukocytes and rouleau formations here of all the red cells clumping together. Sickle cell, genetic disorder where the hemoglobin alters its shape. The hemoglobin, because of one mistake in the order of the amino acids, the hemoglobin becomes brittle. The hemoglobin becomes brittle and reduces its ability to carry oxygen by about 80%. When your individual with sickle cell disease begins to exercise or do something strenuous, the hemoglobin starts to clump together. The red cells form this characteristic sickle shape. And again, they can't carry oxygen. When they become cyclic in shape, they also become very brittle. And red blood cells need to be squishy as they go through the tiny capillaries. A brittle red cell is going to be painful. And so you see this, this characteristic sickle shape here, this curved shape. It It is a homozygous recessive disorder. You have to have sickle cell disease is a recessive trait. You have to have both recessive traits from the mother and the father to have the condition. If you have just one trait, one gene for sickle cell disease, then you're a carrier, and you may have no symptoms. You may have very mild symptoms in here, and so we see this. You see lots of normal red cells, but you also see a lot of these abnormally shaped sickle cell red cells. The problem, of course, occurs when your patient tries to do anything strenuous or active, and that's when the depleted, the inability to carry enough oxygen really becomes a problem. So treatment works very well. If you have a bone marrow transplant and you kill off all the bone marrow that produces the defective red cells, and you transplant in healthy red cells, healthy bone marrow, healthy stem cells, and they will continue to produce. They'll produce new red cells that do not have this disease. The patient is still, it doesn't affect the genetics. A sickle cell patient that has a stem cell transplant, bone marrow transplant, is still carrying both genes for sickle cell. They can pass it on. You don't catch sickle cell, it has to be inherited. So, but at least they're not going to have the health issues if they're producing new bone marrow. So, anyway, more about this in lecture. And that is human blood. So let me get out of here. I'll go ahead and post this for us, and we can stay current.